be testing the myth that the younger you are, the higher frequency you'll be able to hear.
So we're testing this by testing the hearing of 4 age groups, 4 to 6, 12 to 13, 16 to 17, and 40 plus. What's your name?
Grace.
How old are you?
Look at night. One, 2, 3, 4, 5, 6. I am 4 years old.
Hertz. 10 kilohertz. 12 kilohertz. 14 kilohertz. 15. 16 kilohertz. 19. あ、聞こえるんだ。聞こえる？
難しいと思う。
I'm Angela. I'm 14 years old.
I'm Austin Chin and I'm 12.
8 kilohertz. 10 kilohertz. 12 kilohertz. 14 kilohertz. 15 kilohertz. 16 kilohertz. 17 kilohertz. 'My name is Cindy and I'm 16 years old.
Hi, I'm Rena and I'm 17 years old.
My name is Isaac Choi, and I am 19 years old. Yes.
This is 8 kilohertz. 10 kilohertz. 12 kilohertz. 14 kilohertz. 15. 16. 17. 18. 19. And 20.
I'm Akiko. I'm 45 years old.
Fred Choi, 49.
My name is Tim Chen. I'm 47 right now.
8 kilohertz. 10 kilohertz. 12 kilohertz. 14 kilohertz. 15 kilohertz.
就听得对。
哦，那你就
那你就听。
So, what causes younger children to hear higher frequencies than adults? Well, as people age, their mitochondrial DNA can change drastically, and this can have unpleasant side effects. Let's start with the basics. When sound waves travel through the canal of our ear, they hit the tympanic membrane or eardrum and cause it to vibrate. This vibration prompts movement in the ossicles, which transmit the vibrations to the oval window. This essentially creates fluid in the cochlea to travel in a certain way, depending on the frequency of the sound waves that enter the ear, causing small waves within the basilar membrane itself that travel down the membrane. As the membrane gets pushed back and forth as the fluid moves into the cochlea, ultimately causing specific hair cells sensitive to the movement of the waves to activate. The hairs on these hair cells are called stereocilia, and they move as the basal membrane vibrates, and when these hairs move back and forth, the hair cells are activated. The basilar membrane is a membrane within the organ of Corti within the cochlea that contains what are known as hair cells. Different sections of the basilar membrane respond to different frequencies of the waves that travel through them. The hair cells at the very base of the basilar membrane of the cochlea are activated by very high frequency sound waves that reach the ear. Hair cells at the very apex of the basilar membrane of the cochlea are activated by low frequency sound waves that reach the ear. In other words, the hair cells in the basilar membrane are attuned to the particular frequency. This way, the basilar membrane accurately translates the frequencies of the sounds picked up by the ear into representative neural activity that can be sent to the brain. When the specific hair cells sensitive to the particular frequency of the sound waves that have entered the ear are activated, they will fire an action potential. Specifically, the ion channels open up and cause the release of neurotransmitters to propagate the auditory signal to the cochlear, the vestibulocochlear nerve, which will carry this auditory stimulus to the brain. And once the signal reaches the brain, it is to be analyzed and interpreted as a specific type of sound. This signal will reach the brain and will be mapped into a very particular part of the brain. Different parts of the auditory cortex receive information from hair cells that are sensitive to different frequencies. Mitochondria are organelles within cells that convert food into a form of energy that cells can use. The mitochondria contain a small amount of their own DNA. As mitochondrial DNA accumulate more and more mutations, this can drastically change the function of the hair cells in the inner ear and cause them to malfunction and ultimately die. As people age, mitochondrial DNA accumulates damaging mutations including deletions and other changes. This damage results from the buildup of harmful molecules called reactive oxygen species. Reactive oxygen species are regarded as toxic products of cellular metabolism and can function as signaling molecules that regulate many physiological processes. As a result, their buildup can cause the death of irreplaceable hair cells. Cells that have high energy demands, such as those in the inner ear that are critical for hearing, are particularly sensitive to the effects of mitochondrial DNA damage. Mitochondrial DNA damage in the inner ear can be detrimental as it can cause hair cells in the cochlea, which act as sensory hearing cells, to die or become damaged. These hair cells, as you recall, are responsible for translating sound waves that enter your ears into electrical impulses, which your brain interprets as certain types of sounds. The outer hair cells toward the bottom of the cochlea, closer to the opening of the ear, are impacted first and are more likely to be degraded than the inner hair cells located near the top of the cochlea as mutations accumulate. And because of this, as an individual loses more and more hair cells, they lose their ability to hear sounds of higher frequencies before they lose their ability to hear sounds of lower frequencies. Long-term exposure to sound is also responsible for the fact that adults are less and less likely to hear higher frequencies as they age. The outer hair cells are much more sensitive to noise exposure than the inner cells, so outer hair cells degrade first, and as a result, adults first lose their ability to hear sounds of higher frequencies before they can lose their ability to hear sounds of lower frequencies.